So it's a real pleasure to be here. I'd like to thank the organisers for inviting me to share with you the journey that I've been on in Cork, in the very south of Ireland, along with my colleagues, focusing on the role of the microbiome in brain health. And I'm going to talk about stress and neurodevelopment in particular today as the key aspects of what we do. And I work in a medical school, and everything in medicine comes back to Hippocrates. And Hippocrates is reported to have said, although it could be fake news, that all diseases begin in the gut. And that's quite surprising. If you talk to neurologists or psychiatrists or anyone interested in the brain, they would think that there's something very weird about that statement. So hopefully we can convince you over the next 45 or 50 minutes that there is something in this overall. And with my neuroscience colleagues, they often are very surprised that when I'm interested in the neurobiology of emotions, as I am, that I'm fixated on what's going on in the gut. And, but all we have to do is look to our language. We use phrases like gut feelings, gut instincts. We're gutted when we're disappointed. We make gutsy moves when we're brave. And we have butterflies in our tummy when we're nervous. So perhaps there is an underlying biology to these phrases that we use. And my research has evolved and changed over the last 2 decades, but perhaps the one thing that has remained consistent has been trying to understand the impact that stress has on our brain and stress has on our body in general. And we are living in quite a stressful world, and it's hard to imagine in this lovely campus out here outside lovely Cambridge that things are quite stressful, but my desk on a good day does look like this, and everywhere we go we're dealing with chronic stress overall. And all we have to do is turn on our televisions and we get more and more stressed all the time. And one of the worst types of stresses is that anticipatory type of stress and anxiety stress. And so looking into the future, we also can get quite stressed because Ireland is looking a bit lonely out here in this redrawn map of Europe, shall we say. So one of the key messages that I have for you is that stress, and I'm talking about chronic stress or traumatic stress, is a whole body syndrome. It doesn't just affect a few neurons within the hippocampus in the brain, and we study these neurons, these are fabulous neurons, but it affects our immune system and how it talks to the brain. It even affects our gut barrier function and driving a pro-inflammatory phenotype. And so once we start realizing that there can be such a broad aspect of stress on the whole physiology, then we start thinking about it in a little bit different. And like all disorders, we require animal models to study them in depth, and stress-related disorders come with their own challenges. And we have to kind of admit these challenges. And I like this cartoon from the far side because it does highlight the difficulties we have when we're trying to model complex human psychopathology in a mouse or a rat. And we can't just put a rodent on a couch and ask it about its childhood or whether it has recurrent thoughts of death or suicide. So acknowledging these drawbacks in the models that we use, we also can come to the realization that many of the circuits in the brain, for example, that are underpinning the endophenotypes of these disorders, like the fear circuits through the amygdala, the memory circuits in the hippocampus or the reward pathways, have been evolutionarily conserved. So we can use rats and mice with some level of confidence that they'll tell us something about the human condition. And many moons ago, we were, and still are, interested in stress in the perinatal period. Because this is adverse child events are really associated with many stress-related disorders, and we were interested in trying to model this in animals. And Siobhan O'Mahony, who was a PhD student at the time, started working on this early life stress model in rats, where rat pups were separated from their moms from postnatal day 2 to 12. When they grew up, these animals have a whole body syndrome with changes in their immune system, in anxiety, in depression-like behavior, in their gut barrier function, in their stress responsivity, in their brain chemistry, even in visceral pain, a core symptom of irritable bowel syndrome, were all changed. And that's what we would have predicted. But then Siobhan was collaborating with Julian, indeed, when he was in Cork at the time, using very crude techniques that are almost embarrassing to show at a meeting like this. But they did make a point in that we saw a reduction in the diversity of the microbiome in these animals that persisted into adulthood. That there was a signature of this trauma that could be detected in the microbiome. Now, this could have been epiphenomenological, it could be due to many things, but it got us thinking, well, what if there's something in this, and what could it be that stress affecting the microbiome could be part of how stress is mediating its effects? Just interesting that in the last week or so, a paper came out in Humans from Neil Epperson's group when she was in Penn. They showed that something similar, that there was a persistent signature of early life adversity that persisted in moms all the way through to their own pregnancies and was able to moderate how they responded to stress during pregnancy. So we're beginning to see translational aspects of this. We then went on to look at prenatal stress using better techniques, and we were able to show that even stressing rat moms during pregnancy, that in the offspring, the microbiome is also different and that it correlates with changes in physiology. And again, as a human translation of this is this study from Willem de Vos and Carolina de Verts from Utrecht, where they showed that in moms with high perceived stress during pregnancy, and first-time moms, pregnancy can be quite stressful, that there is a signature of this stress in the offspring's microbiome. So altogether, what we're seeing is that there's a very close interlinking between stress and the microbiome, and that's what set us on our journey that we've been on ever since. Now, most of you here, I don't need to tell you about the microbiome, etc., but there are a few things just worth reinforcing in terms of numbers that one never gets tired of hearing. First of all, like in terms of genes, we are more than 99% microbial. And, you know, we're here on this campus, and that's quite humbling to think about. It's also quite humbling as a neuroscientist to think that the weight of our gut microbes is roughly about the same as our brain. And this is a very fast-moving field with things getting changed all the time. So if I'd given this talk a few years ago, and it's still well used in the literature, I would have said we have 10 times more microbial cells than human cells. But this has been revised downwards, so it's about 1.3 to 1. And so we are still more microbial, but just about. And as I like to say, just think that the next time you go to the bathroom and you get rid of some of your microbes, that you're becoming more human, and what that might mean overall. When we talk about the microbiome, again, this is a lot of people here know this already. We often just mean the bacteriome, and we neglect the other components of the microbiome. And there's lots of really exciting work going on, and you'll probably hear a lot of it at this meeting in terms of viruses and bacteriophage, archaea and fungi. And so we can't neglect this. And I don't need to tell you how popular it is, making the cover of all the glamour magazines, and how much the lay press love it. What we've been interested in from a stress perspective is really this saying from Hans Selye, who's the father of stress research, trying to understand that he said that it's not stress that kills us, but it's our reaction to it. So why on this roller coaster of life, if two people are subjected to the same insults, could they respond differently? Why is it that when we all turn on our TVs that we don't have a stress-related disorder? And we know that genetics is really important in this. We know the growing field of epigenetics is also really important. But also we began to start thinking about, well, what if the microbiome is also playing a role in this? And a lot of focus in stress research has been on stress susceptibility. But over the last number of years, people are beginning to understand what are the resilient factors that are protecting us from this. And that's something that we're really just beginning to tackle at the microbiome level now. And we're doing this again again, in animal models to start with, but we also have some human parallels to look at. And we use a model of social defeat stress in the mouse, and we're beginning to look at what's going on in the brain, what's going on in behavior, and you can see that certain animals, and these are genetically identical, fed the same diets in a very controlled environment. And this is the beauty of these animal studies, because they allow us to control so many things to try and get at some mechanisms. So their behavioral and physiological response to stress will vary. We're looking at using RNA-seq to see what's going on in the brain, and then we're trying to also look at what's going on at the microbiome level to see if there's anything that we can predict in terms of how the microbiome responds to stress. And again, this is early days, but what we find is that when we stress mice, and we look at changes in the microbiome, in some of the mice there are really big changes, and the microbiome is quite volatile, and in others they seem more resistant to this. And so the questions remain: what determines the degree of this volatility overall? And moreover, can we predict, are there specific keystone species at baseline that will predict whether certain animals will respond or not? And as I said, this is we're at the beginning of trying to tackle this, but I think it will be interesting in the future to be able to look at larger human cohorts as well to see if there is stress susceptibility factors within the actual microbiome. As I mentioned, we're interested in stress in early life, but we're also interested in stress across the entire lifespan. We have programs in the adolescent period and in aging, and I'll talk a little bit about aging as well today, and trying to understand the relationship between stress and the brain at these key critical windows and how the microbiome shapes it. Of course, we have to think about where does it all begin, and while it's largely thought that that we are sterile in utero, factors that can influence the mom's microbiome during pregnancy can have a long-lasting impact on what is transmitted then and handed over to the infant at birth. And in a way, I like to see this handover from mom to infant of the microbiome like some kind of evolutionary relay race. And with the microbiome, the vaginal microbiome serving as a baton to prime for future health overall. And so factors that influence the vaginal microbiome, such as antibiotic exposure, a lot of really exciting work from Tracy Bale's group on stress, on infection, on varying diets, will influence what is handed over. Then in the postnatal environment, of course, these same factors will also play a key role, with breastfeeding and mode of nutritional provision being very important in shaping the postnatal environment. And then throughout the lifespan, a variety of factors, many of which you'll hear about at this conference, will influence the microbiome composition. But one of the things we've been interested in is if an infant is born then by C-section, things would be different, because the vaginal microbiome isn't handed over. And this goes back to early work from Gloria Domingos-Bello's group, where they show that C-section born individuals have a different microbiome than those born vaginally. Now, this is a very small study, almost pilot-like in its fashion, but it has been reproduced now in 14 different cohort studies. There are intriguingly 2 negative ones, but for the most part, and this is exemplified in this study that came out a few weeks ago in Nature, where it showed that birth mode was a very significant factor in determining the microbiome in infants that they followed up, and quite a large infant cohort that they followed up over time. Moreover, in this paper, the breastfeeding was also very clearly a factor in early life that would determine the microbiome. So if C-section is so important in terms of disturbing the normal laying down of the microbiome, then we were interested to see whether there are any long-term functional consequences of this on the whole system and on the stress response in particular. And we know that infants that are born by C-section have an increased relative risk for allergy and asthma and type 1 diabetes. We know very little in the context of brain health. And so that's something that we were working on. We've developed an animal model of C-section that we've been working on now for many years. And we find that indeed, in this animal model, there is a disturbance of the microbiome in early life that starts to come together by adolescence, and in adulthood, you can't distinguish C-section from controls. And intriguingly then, when we look at behavior and physiology, we find that there are deficits and enduring deficits in anxiety that can be detected as early as postnatal day 9, that persist into adulthood and that are combined with deficits in social behavior as well. And so that tells us that there are these long-term effects. And we find that these effects can be ameliorated if we target bifidobacteria in early life in particular, and that's something that we're also quite intrigued about. But that's mice, and many of you here are human researchers, and you feel that there's many drawbacks to mouse models in general that I alluded to. So then we wanted to see, is there a way that we can ethically stress people in the lab? And my PhD students would say they're doing that all the time. But we have a very well-validated task called the Trier Social Stress Test, which is good for inducing acute stress. And so this is where someone has to give a public speech where they have to petition for their dream job with two angry-looking people looking at them and a camera on them, and you mount a really good psychological and physiological stress response. And so we took, I was going to say normal, but Irish medical students, and we decided to stress them, and what we found was and segregated them by their mode of delivery, because most people know how they were delivered. And what we found was that there was an increased immune and psychological in particular stress response in those born by C-section compared to controls. So that's acute stress. But then what about chronic stress? Because that's really more interesting in many ways. And so then we were faced with a challenge: how do we ethically, chronically stress people? And that's the great thing about being an educator, is that we do this all the time by subjecting people to exams. And so we look at the chronic examination period, and we're able to look at how people stress response changes during that. And intriguingly, and this is, we didn't expect, because there's so many confounds that could be in this type of study and type of analysis, but it really builds on what we've seen in the animals, is that there was an increased psychological response to stress in students that were born by C-section compared to controls. And this is the Cohen Perceived Stress Scale. Those that were born by C-section really seem to have a much greater response to the examination period. Now, this could be more than just microbiome disturbances. The stress of being born is taken away, the immune priming, many other things. But it is intriguing, and it just is a demonstration of some of the translational approaches that we're trying to dig at and get at some of the mechanisms that are underpinning this overall. And the important thing also to realize is that with C-section rates, going skyrocketing, especially in developing countries, in parts of China, for example, it's at 60%. In parts of Brazil, 70%. Even in Ireland, it's doubled in 30 years, the C-section rates. So, but the last thing I want to be here is a male talking to people about their obstetric choices, but and sounding that this is a really bad thing. C-section is a life-saving procedure, and the WHO would say about 14% of all pregnancies should be C-sections to give optimal health for both the mother and infant. And so the question is, if people have to have a C-section, is there anything that we can do to try and reverse this microbial change in composition? And that's something that we're also working together with some of our industry partners, but also to try and look at dietary mechanisms that could impinge on supporting the optimal development. And I mentioned bifidobacteria earlier, but using prebiotic or probiotic approaches to try and recover some of these deficits, and that's something that we're working on at the moment as well. So, summarizing the early life microbiome brain link, we're beginning to expand the Barker hypothesis, which is this developmental origins of health and disease, which is very much has three pillars to it: the prenatal environment, the postnatal environment, and host genetics. Well, now we need to add the microbiome into this in relation to understanding it and bringing it to a four-pillared developmental origins approach, and that are each are playing a role in shaping neurodevelopmental trajectories in early life. And we'll hear more in the session tomorrow from Rochelle and others about this. So switching to the other extreme of life, to aging. We're also very interested in how the aging brain deals with stress, because what we know is in aging, there's a process called inflammaging happening in the brain, and basically an inflammatory state releases lots of negative molecules that can very much damage the brain, and when you add stress to that, you really are adding fuel to a fire. And what we know is that stress-related disorders like depression and anxiety are particularly difficult to treat in geriatric populations. We know, of course, that stress will drive the cognitive impairment, and social isolation and the whole concept of loneliness is only now beginning to get proper attention from a public health perspective as well. And I will just mention that this paper from colleagues in Cork, from Paul O'Toole and Marcus Clayson from some years ago in Nature, where they showed that the microbiome was much less diverse in elderly individuals, and that this diversity correlated with health outcomes. So that the people that had the better indices of frailty, for example, had the more diverse microbiome, and what was driving it all was in terms of the diversity of diet. So with these types of studies, we're beginning to see we don't just have a gut-brain axis or a microbiome gut-brain axis, it's really a diet microbiome gut-brain axis. And that's really what's informing a lot of our thinking moving forward, is using dietary interventions to try and shape the microbiome at critical windows, either in early life or in aging, to be able to drive positive health outcomes overall. And many other people are working on the microbiome and aging, and in the whole aging field, this is a study from Italy on extreme longevity, but in the whole aging field, the microbiome is one of the hottest areas there is right now, whether it's in the context of frailty or immune health or gut health or cognitive health, is what we're interested in general. And while it's really exciting and really new in aging, the other thing about this field and science often in general is that there's very little that's really, really new. And so if we had listened to what Melnikoff had told us, we would already know that there was a strong link between aging and the microbiome. So Eli Melnikoff was a Russian working in the Pasteur Institute in Paris, and he won the Nobel Prize in 1908 for his seminal work on discovering how macrophages work and phagocytosis. And perhaps like many great scientists in their later years, he came up with crazy ideas. And he was full of crazy ideas. And one of his crazy ideas was, why did people who lived in parts of Bulgaria, what's now Bulgaria, why did they live longer? And he wrote extensively about it. And he put it down to the fact that they ate a lot of fermented foods containing lactic acid bacteria. And so he's not only the father of one of the key fathers of immunology, he's one of the fathers of geriatric science and trying to really look at healthy aging, and also one of the fathers of probiotic research. And if you go to Korea, for example, you can get a yogurt drink which has his picture on it, so it has that Nobel sign of approval, so sure, it must be good for you. So the thing with Melnikoff is that most of what he wrote about was forgotten about for 70, 80 years, and it's only in the last few decades that people have begun to realize how much was in what he was writing about. Recently, we've been revisiting some of these concepts of Melnikoff in the context of gut-brain health, and we've shown that in elderly mice there is deficits in social behavior and anxiety and cognition, and we're trying to look at some of the mechanisms that are driving this. And what we find is in elderly mice there's a increased intestinal permeability, but when we stress them acutely, it even drives this much more. We begin to look at the inflammatory mechanisms, and we see an increase in pro-inflammatory cytokines, as you would predict, and also that there are changes at the microbiome level. And now what we're trying to do is then, can we target the microbiome in some ways to reverse some of these effects? And this is part of a European project, and we decided in this project to focus on middle age, maybe because that's something that is I'm very interested in right now, shall we say, and also because we feel that in middle age is a good time to start to change brain trajectories. And in this project, we looked at whether a prebiotic, in this case inulin, could reverse some of the neuroimmune changes that we find in aging. And in these middle-aged mice, there's an increase, for example, in stress-induced immune priming, and when we pre-treat with this inulin, we were able to actually dampen down this. Now, microglia are basically the brain's macrophages, and they are really important in shaping neuronal activity, and they become hyperactivated in inflammatory states and in neurodegeneration. And what we see in aging is that there's an increased microglia activation, but if we pre-treat with a prebiotic that's targeting the microbiome, we can reverse this, and we can also reverse many of the changes at the microbiome level. So we're beginning to look at ways that we can promote healthy brain aging and neuroinflammatory responses through dietary targeting of the microbiome. We also have an ongoing project now to see if we can, by actually transplanting the microbiome from young to old, whether we can reverse some of these types of effects as well. So that's the journey that we've been on, but my neuroscience colleagues are a hard bunch to convince. So they still are, it's a lot of correlations, and it's all over the lifespan. So really, John, is the microbiome playing a key role, yes or no? And in biology or in engineering in general, probably the best way to find out if something is involved is to take it out and see what happens. And so the concept of being germ-free, living without microbes, is nothing new. It goes back to Pasteur's time. And you can see here is a germ-free man from a sci-fi magazine from the 1920s. Our germ-free mice look a good bit better. They have their own problems, and we're very fortunate in Cork to have a germ-free facility. And these mice, they grow up in a bubble. They lack any real clinical translation, and that's one of the drawbacks of what we do, with maybe the possible exception being the boy in the bubble that Paul Simon sang about in the 1980s. But they've been a very useful tool for us to understand the relationship, whether the microbiome is involved, yes or no, and to start us on trying to deal with mechanisms. And all of this goes back to a paper from Japan, from Sudo's group in 2004, that was published in the non-glamour journal, Journal of Physiology, which showed that germ-free mice, when you stress them, they have an exaggerated endocrine response, increased ACTH and corticosterone, the main stress hormones. And moreover, when he put back in a single strain, in this case a bifidobacteria, he's able to reverse, and this goes back in post-weaning, this reprogramming of the HPA axis that has occurred. So this paper came out and was more or less ignored for many years. And then we were really interested when we found that stress was affecting the microbiome in our early stress models. We started thinking, well, maybe these germ-free mice have also got changes in their brain level. And we were very excited about this idea, but any time in science when you come up with something that's really exciting and new, it's of course not unusual that other people also think of the same things and publish it before you and in different ways. this is what's happened in this regard, because Rochelle Diaz-Heiss and her team in Karolinska published this really nice paper which really changed a lot of the concept of neurodevelopmental world to really focus on the microbiome, followed up by work from Jane Foster in McMaster, and then later our own paper to show that the germ-free mouse has deficits in neurodevelopmental trajectories, changes in anxiety behavior. And what's really nice about all of these studies, and there's been more since, is that there's lots of convergence overall in terms of what we see. There are differences as well, but on the whole, what we're finding is that if mice don't have microbes during early life in their gut, the brain doesn't develop properly. That's a simple answer. Here's just some data. Brain-derived neurotrophic factor is a key plasticity-related protein, very important for learning and memory. You can see there's a deficit in germ-free mice versus control. We look at serotonin, a very important molecule involved in emotion, and that's increased in the hippocampus. And then we decided to look at more sophisticated elements of neuroscience in the hippocampus. And one of the things we wanted to look at was the birth of new neurons, which happens throughout our lifespan. And so we looked at adult hippocampal neurogenesis, and we can label them here with BRDU, and we found that actually there was an increased neurogenesis in these germ-free animals compared to controls. We then turned our attention to another brain area, the amygdala. The amygdala is the brain area involved in fear, in the social behavior, in the emotional aspects of pain. It's this almond-shaped part of the brain. And you don't need to be a neuroscientist to be able to see here very clearly that there is differences in the arborization and the branching of this basolateral amygdala neuron in germ-free versus control. We've tried to look at what does this mean from a molecular point of view, and using unbiased RNA-seq approaches, we're able to determine molecular pathways that are different in the germ-free versus control animals. And we've also been focusing on microRNAs, which are key players in the epigenetic landscape of regulating gene expression, and they are also quite different in the amygdala and in the prefrontal cortex. We then turned our attention, and in terms of what does this mean functionally to the animal, well, then we looked at certain amygdala-dependent behaviors, and the ability to associate fearful responses is one of these. And we find that in germ-free animals, there are deficits in how a mouse is able to remember an association between getting a foot shock and the context it gets it in. And so basically, the animal is not freezing to the same level. And what was interesting, when we put microbes back post-weaning in these animals, we could recover some of this phenotype. I mentioned pain, and what we also find is that there's an increased visceral pain response in these animals as well. And what was also nice here was that there is plasticity in the system. Also, when we put microbes back post-weaning, we can reverse this phenotype. So again, really conclusively showing us a link between core symptoms of stress-related disorders like irritable bowel syndrome and the microbiome. We then turned our attention to the prefrontal cortex, which is the front part of the brain, which is very much involved in higher cognitive function. And we did a same RNA-seq approach, and something we never expected to come out came. And that was that a lot of the genes that were upregulated were involved in the process of myelination. And myelin is the insulation in which nerve cells require to have optimal conductance. And you may know of myelin from hearing about demyelinating disorders like multiple sclerosis. But what we found in our mice was that, especially when we looked down the electron microscope, was that there was a hypermyelination. And it was only happening in the prefrontal cortex, and it was mainly happening in males, not females. So these were quite intriguing because hypermyelination doesn't occur that often in nature. It's a few rare autisms where it has been shown to be found. The specificity of the prefrontal cortex is intriguing to us, and the fact that it's, like in all of our germ-free studies, much more robust in males than females is quite interesting, because as a stress researcher, many of the readouts you have are much more prominent in females than males. And so that got us really into thinking about autism, something that we hadn't been really working on before that. And it's also not well discussed, the relationship between GI distress and autistic behavioral symptoms. And now we realize that there's a large percentage of autistic kids and autistic adults have functional bowel problems that are very difficult to manage. And so we wanted to look at some of the core symptoms of autism in our germ-free mice. And mice, like humans, are quite social. So if you give them the opportunity to spend time with a mouse or an empty chamber, they'll gravitate towards the mouse, but not if they're germ-free. Mice, like, well, some humans, can be a bit fickle. So if you give them the opportunity to spend time with a new playmate versus their everyday playmate, they'll prefer to spend time with the new playmate. But again, not if they're germ-free. These germ-free mice also have increased repetitive behavior, so that's quite intriguing to us. More recently, we've been trying to map out some of the molecular pathways by forcing interactions and forcing social interactions in these germ-free animals. And what we find is in normal animals, in grey here, that we get an activation of the MAP kinase signaling pathway in the amygdala that's severely attenuated in germ-free animals. And on the other side, in the germ-free animals, there seems to be an enrichment in genes involved in alternative splicing. And we've been trying to tease apart the pathways that are affected by this. And this is just the beginning. It's only scratching the surface of what's going wrong, but it's telling us that there are changes that are driven in early life by the microbiome that are shaping neurodevelopmental trajectories and shaping how the amygdala responds in general. We've taken the other approach, which is to take, what if we took a mouse model of autism? In this case, a genetic mouse model called the BTBR mouse. These mice have impaired sociability, enhanced repetitive behavior, impaired cognition, and high anxiety. And we decided to look at, well, what's going on in their gut compared to normal black 6 mice? And what we find is there are deficits in terms of bile acid metabolism and tryptophan metabolism. And this is work we did in collaboration with Susan Joyce and Cormac Gahan in the APC. And this really helps to explain many of the functional GI problems that we see in these mice, such as delayed motility and increased permeability. And the questions remain is if we can correct some of the gut problems in these mice, could we ameliorate any of the behavioral symptoms? And work is ongoing to try and tease that apart right now. In line with this is an emerging field from other groups which are working on mouse models of autism, where they've shown that the microbiome is disturbed, and usually in a sex-dependent fashion, mainly in males, as you would expect. And this is showing that in mouse moms that are given either the drug valproate, which has clinically been shown to increase the risk for autism in offspring, as has maternal inflammation or maternal obesity, all of which in epidemiology studies have been validated to increase the relative risk of autism. And in these mice models, they also increase the behavioral changes that are akin to autism. Really intriguingly is the realization from the late Paul Patterson and Sarkis Mazmani and Elaine Chow, where they gave a single bacteria, a Bacteroides fragilis, in early life, or in Mauro Costamaggioli's work, where he gave a Lactobacillus reuteri in early life, and then more recent work in a genetic model from Israel, where they also give Lactobacillus reuteri in early life, and they could reverse a lot of the behavioral changes. So this is telling us that we could potentially intervene with a microbiome targeting approach to try and ameliorate some of the symptoms of autism, if this could translate to humans. So that's the big if right now overall. It's also worth noting, if you look across species from an evolutionary perspective, that when we look at the relationship between the microbiome and behavior in a variety of species, from locusts to termite to honeybees, zebrafish, birds, the koala, hyena, all of these have studies linking the microbiome and ethologically relevant social behavior. So there's something about the microbiome and social behavior, which is at the core of autism, that it seems to be intriguingly linked. And that's something that we've been quite interested in for a long time. And some years ago, together with Roman Stilling, a postdoc in the lab, we started thinking about really triangulating what we know in terms of, we know the microbiome now is involved in brain development, we know the microbiome is involved in social behavior, we also know based on the Dunbar theory that the social behavior has, in evolutionary terms, has helped the encephalization or the growth of the frontal cortex. So you know, we start thinking about, well, if we live in social groups, what's in it for the microbes? Could it be good for microbial fitness? And this is some of the ideas that I put to Seth Bordenstein, who's an evolutionary microbiologist in Vanderbilt, and he told me that I was completely wrong. And every now and then, as a PI, it's good to be told you're wrong, and that's why we need collaborators who are experts, and that's how we need to value cross-disciplinary fields. And the reason that I'm wrong is that I'm forgetting that the microbes were there first. And there never has been a time where the brain has existed without microbial signals. And we can't just separate, well, what's in it for the microbes. It's what's in it for the entire holobiont overall. And so we begin to really, when you start thinking like that, your view on brain development and how the microbiome is shaping it changes in different ways. And that these are really our friends with social benefits and many other benefits as well. So I mentioned that stress, especially in early life, will change the gut microbiome. When you take out the microbiome, you alter the stress response and your developmental trajectories. And that leads us to the question, could we modulate the microbiome and have beneficial effects on brain health? And this slide summarizes loads and loads of work and many, many PhD theses, because it's just showing you the positive data that we found. And we found that specific strains of bacteria, certain prebiotics, and now more recent work together with Paul Cotter's lab in the APC on fermented foods, that they can have beneficial effects in mice on anxiety, on depression-like behavior, on cognitive processes, and enhancing the stress response. They can modulate brain chemistry, especially in GABA and serotonin in the brain, and this has been really exciting. But again, I need to reinforce that many, many strains of bacteria are doing nothing overall, and even some prebiotics we've worked on have not given us any benefit yet. And so understanding why certain bacteria are beneficial and others not is really going to be important in really determining the molecular mechanisms as to how microbes are affecting physiology and brain in general. And that's really where we need to go, is try and understand is how do microbes in the lumen of the gut signal to the brain to induce complex behaviors. We know that the enteric nervous system is very important, the gut barrier function is very important, the immune system is playing a very vital role. But I guess one of the things, and the vagus nerve I'll come back to, but one of the main things to reinforce is that the microbiome is like a factory producing all kinds of weird and wonderful molecules that our bodies wouldn't have without them. And so short-chain fatty acids are among the most studied in this regard, and we wanted to try, well, what happens if we bypassed the microbes and went straight to the short-chain fatty acids? And so in a study, this is work from Marcel van der Waal, a PhD student in the lab, where we were able to show that chronic stress induced many, many changes in physiology and behavior. And if we co-treated with a cocktail of short-chain fatty acids, that we were able to ameliorate the effects on anxiety, on gut permeability, and on the stress response. Many factors weren't changed, but it does tell us that short-chain fatty acids may be responsible for some of the beneficial effects of certain pre- and probiotics in general. I mentioned the vagus nerve. This is this long wandering nerve from the brain to all the organs, including the gut, and it's bidirectional in terms of its communication. And some years ago, together with John Bienenstock's group in Canada, we wanted to see, does it play a role in how Lactobacillus rhamnosus is mediating its effects. And so we cut the vagus, and to make a long story short, all of the behavioral and neurochemical effects that we'd seen were now gone. So this tells us that what happens in vagus doesn't just stay in vagus, but will actually affect our emotions in certain ways. And that's been quite interesting. And this paper came out today. So it's in Neuron Today from the Costi-Mattioli's group, where they showed that basically an L-Reuterie could reverse the autistic-like behaviors in a genetic model of autism, and when they vagotomized their animals, the effects of the L-Reuterie were gone. So again, reinforcing the importance that the vagus has as a conduit for signaling from the gut to the brain. But it's only one of the pathways. It's not going to be the whole story, but it is an intriguing story. And more studies need to be done to really delve into the mechanisms. Maura's group also focused on oxytocin as a key mediator here as well, and that's something that we're also interested in because oxytocin is a key molecule involved in social behavior in general. So no microbiome talk or plenary is complete without at least mentioning fecal microbiome transplantation, taking someone else's poo. An ancient practice going back to 4th century China, and it is basically disrupting medicine in many ways. Changing what we think about medicine and how we deal with it. And so people are looking at new ways to innovate. These are the capsules that have been generated to try and deliver this medicine in innovative approaches. There's very little on it in regard to brain health. There's one pilot study in autism which has shown some beneficial effects, but it is a pilot study, but they've shown that FMT can actually alleviate both the behavioral and gut symptoms in autism. We were interested in using this FMT approach, and so we looked at the microbiome of depressed individuals versus controls, and then we took that microbiome, because we showed that it was a reduced diversity, and we transplanted it to a rat, and we were left with a rat that had depressed-like symptoms. And these were manifested in terms of anxiety, anhedonia, and increased inflammation. And so this helps us to start to move away from just correlation towards causation and try and see what are the mechanisms in mediating this. But it does tell us that there's something in the microbiome that it has the potential to affect brain and behavior in different ways. And all of this brings us to the whole concept of psychobiotic. It's a term that my colleague Ted Dinan came up with, and we, in this book for the lay press, or lay public, we predict that we're just at the beginning of a psychobiotic revolution and trying to understand this relationship between diet, microbiome, gut-brain axis overall. And we've more recently expanded what we mean by a psychobiotic with colleagues in Oxford to basically be any targeted intervention of the microbiome that will support brain health and behavior. But do we have any evidence that is there anything in this idea in humans? Because all of what I've told you up to now has been really founded in basic neuroscience work in animal models. And indeed there is. This is a small study, so it's only 22 participants, but it was quite an interesting study, a placebo control study, where we looked at a specific bacteria that came through our preclinical pipeline. So it's a Bifidobacteria longum. It's shown efficacy in our animal models, and then we gave it to healthy volunteers for a month. And we were able to show that when we stressed them in the lab, that their stress response was greatly attenuated. We looked at their EEG responses, and we could detect that there was an EEG, differential EEG response, and we also looked at cognitive function. And this was really, really exciting for us because it's telling us that some of the work we're doing in animals is able to go the whole way to humans and be validated. And so then we decided, let's take our most robust bacteria that we've shown the most data in a preclinical setting and tested in humans. And we did. And you cannot get more negative data into the one paper if you tried. It did nothing. And so, this is just to highlight some of the limitations that we have in all fields. It's not specific to the microbiome field, or is in trying to translate, especially from healthy volunteers, studies in animal models. And we have a long way to go, and we need to do a lot more, because these were healthy people. So it may work if we went to a different population. There are more and more studies emerging, and they're getting bigger and bigger, thankfully. So we're going to see a lot more in the coming months months and years showing both negative and positive studies in the field. And the field needs plenty of negative studies as well as positive studies to understand what is the true signature of a psychobiotic in this regard. The last thing I want to mention is something we're also interested in right now is the impact that medications and drugs have on the microbiome, and in particular drugs that we take for brain diseases. We've shown many years ago that antipsychotics increase weight gain and change the microbiome. We're able to attenuate this weight gain by targeting the microbiome with antibiotics. And then Sofia Cosoto decided to broaden out the repertoire of drugs that we did. She looked at 7 different drugs, both in vitro, on gut permeability, and in vivo. And took her almost a year of her life doing this with these 7 drugs, and found really intriguing findings on the microbiome level and on gut level. And then this paper came out, which had over 1,000 drugs. So you can imagine the PhD student that day when someone is showing over 1,000 drugs. And this is from the Embl group in Heidelberg. But it was really interesting that they did show that antipsychotics were overrepresented in terms of drugs that were affecting the microbiome, and that about 1/4 of all drugs that are in pharmacies will modify the microbiome, and many of that is in a negative way. So this is a very exciting field where you can tell I'm very enthusiastic about it, but we have to be careful about where we are on this hype cycle, especially in relation to brain health. And we all will know about Jonathan Eisen, and he's overselling the Microbiome Award. That's not something you want on your CV overall. The field is challenged by the difficulties between going from rodent studies to human studies. I've already mentioned about correlation not meaning causation. And there's a wonderful website called Spurious Correlations where you can go and find all sorts of things. And one of the things is that the divorce rate in the state of Maine correlates with the per capita consumption of margarine. And so this tells you the dangers we have in this field. There's lots of small underpowered studies, especially in the autism field, lack of dietary information and good psychiatric phenotyping, and we need a lot more rigorous placebo-controlled trials and really to get at the mechanism moving forward. But we're really optimistic because we feel that the microbiome is the epitome of personalized medicine into the future. But it comes with its challenges because we don't know what a normal microbiome is, and I'm sure we're going to hear a lot about that over the next few days. We see a lot of advances in terms of longitudinal sampling of the microbiome and citizen science approaches that are going to help with that. We're going to see a lot of focus on personalized diets for brain health. And that's what we're really excited about, and trying to understand the interactions between the host genetics and environment. And with Colin here, I can't forget that it's not all about the bacteria, it is also the bacteriophage, and a lot of work to be done there, and we've started doing that as well as focusing on functional outcomes of the metabolome. But and this is also, in this genome campus, difficult to say that unlike our genome, which we can't do much about except blame our parents and our grandparents, our microbiome is potentially modifiable. And so that's something that gives us a lot of agency over our own health moving into the future. So in the 20th century, and most of the focus on microbial medicine was killing bugs, now we really understand that we have to protect our microbiome and that our state of gut will affect our state of mind. So this is the work of a large team of us in Cork, including a big shout out to Ted Dinan, my clinical colleague, our industry funders and all our funders. And we're back to Hippocrates. Because the other piece of fake news that he's supposed to have said is that let food be thy medicine. So I'm going to change that to say let food for your microbes be your medicine. And I leave you with this cartoon that the Daily Mail, dreaded Daily Mail, made about us. So thank you very much.
Thanks, John. Have we got any questions for John?
There was a paper out in Parkinson's which linked, potentially anyway, linked short-chain fatty acids with Parkinson's disease. If short-chain fatty acids are the be-all and end-all of gut health, etc., things like that, why are they saying butyrate is potentially a bad thing?
Sure. No, that's the Tim Sampson sarcosmanian paper. It's intriguing. It's intriguing because they're working on in that paper. They're working on a alpha-synuclein model in a germ-free facility, and I think it's like everything in the microbiome field. It's going to be more complex, and any of my quick generalizations that I've made need to be qualified and will be as time goes on. One of the co-authors in that paper is a clinician who has data showing that even for Parkinson's, that short-chain fatty acids are actually positive good, but it will also depend on where and across the lifespan they're being modified in general. So I think that paper, we need to see more. It's a single publication at this stage. It's about microglia, which are the brain's immune cells, and how the short-chain fatty acids are priming that. In the earlier paper from the Freiberg group, they basically found in the absence of microbes that the short-chain fatty acids were able to reverse some of the good, showing again that they were good. So it's early days, but it is a really, really important point. And when we're trying to develop, in, for example, the diabetes field or other fields where we're trying to develop short-chain fatty acid-based interventions, we need to keep an eye on this in case there is negative CNS side effects down the road.
Having in consideration your, your data from mice, that the germ-free mice have a normal hippocampal development, do you think the same happens in humans developed by, born by C-section?
So there's very little data, really to stratify in terms of functional connectomics in humans and stratifying them by that. I'm not aware of any study really looking into it in great detail. Our germ-free mice are an extreme situation. What we find in our C-section is much more modest. So, what we're thinking is that there is going to be some aspect of rewiring if the microbiome is different in early life, for sure. But whether it would be at a structural level or whether we could identify it even in a brain imaging scan, I don't know yet.
And the other question would be the timing of the treatment. Do you think that's very important? Because if it has impacts on the development, even the immune system, then maybe early treatment is essential and later treatment has no effect.
Yeah, so in some of the cases in germ-free animals, we are able to reverse some of the phenotypes post-weaning, when we intervene post-weaning, which is quite late. In a mouse, it's at P21. So I was surprised that there was still plasticity left in some of these pathways. I agree that earlier intervention, the better you are. But in some of these studies that they're around, because there's pragmatic problems in handling animals in early life that you can actually stress them and you can have a stress effect added on to the actual intervention you're given. So a lot of the studies are at weaning, so you're not disrupting the maternal pup bond overall. But yeah, timing is going to be everything.
Great talk, thanks very much. I'm a clinician who works in neurodisability, pediatric neurodisability, a lot of autistic children, and I want your opinion on food selectivity in the autistic population and whether it's the microbiome that governs the food selectivity or if it's the other way around.
So in terms of whether they respond to reduction diets of key...
No, I'm saying that a child who has a very restricted diet, is that governing their microbiome, or is it the microbiome who's selecting their food selectivity?
Well, definitely the diet is affecting the microbiome, and so the food selectivity will change the microbiome. But what we don't know is, I didn't go into it at all, but the pathways that we have in the brain for actually food choice could also be governed by the microbiome. There's studies in lower animals, in flies, for example, which really elegantly show that food choice behavior is determined by the microbes themselves. So it's probably a circular argument there, but we need to really drill into that, and we don't have enough evidence yet, definitely not in humans. But it's a very valid point.
Have you partnered with anyone to look at mutagenesis libraries based especially in your B-longum 1714 and the L. reuteri, which had a positive and negative result?
Yeah.
And have you tried to put those through your mouse models at all?
Oh, to knock out specific genes?
To go to mechanism.
Yeah, that's what we want to do. We haven't done it yet. We were interested in another strain that was producing GABA, and we were wondering whether, so we were going to knock out the GABA just to see if that was, we haven't done it yet, but that's part of our plan, and that's what's good about being in Cork. We have people who can do that I can talk to, so that's good.
Yeah, there are a lot of probiotics around, and I think some butyrates are also coming as supplements. And what you've presented, working with the gut, how far do you think we are from having solutions reaching the market? Because the diversity of the subject we are dealing is quite huge.
No, it's a good point. It depends. I mean, like, so the biflongum that I worked on there that came through our animal studies, and we did a small pilot study, and there's a brain imaging study in a German group. That was enough to the company that makes it. They put it on the Irish market, and it's coming on the UK market now. So that was enough for something to get pretty quickly onto a market with some evidence. You know, it needs more robust, larger trials and whatever else. I think the inter-individual responses is going to be really important, and understanding individual microbiomes and how they're going to respond to specific, whether probiotic or prebiotic, are also hasn't been tackled in this regard. It's early days, but it's very exciting in that regard.
So do you think it would, because a lot of probiotics are coming as supplements in the market, do you think it will create some trouble for clinically proved products coming later in the market?
No, because the one thing that our data really reinforces is that a strain is going to be really important. And so we have another study where we've looked at 12 different biflongum strains, and they're all having different in vitro responses. So I think we're going to see, if nothing, the message that we'd like to give, and that's why I like to juxtapose the positive and negative data in the humans, is that strain, getting evidence around specific strains. So just going into a random pharmacy and getting random probiotics will not probably answer anything. So I think, but it's messaging in regard to that I think is going to be important.
Thanks very much. John, thank you for that. That was really good. I just had 2 questions on the short-chain fatty acids. So I work a lot with Crohn's and colitis from a nutrition perspective. Has anyone in your team, because I haven't seen the published paper, so I just have 2 questions. When you provided the short-chain fatty acids, did they also give propionic acid as part of that spectrum of...
Yeah.
You did,
yeah.
okay.
Yeah.
And then the second question was, has anyone in the team looked at colon-administered, butyrate enemas? Because I would see that a lot with colitis clients. We would see amazing outcomes. So I'm just curious, because I would administer butyrate a lot orally, but we never really know, does it get to where it needs to be,
No, I'm not aware, but I don't work in IBD too much, but they're really interested in fiber, and fiber as a precursor for short-chain fatty acids. So I think that's probably where it's going, because a lot of people, eliminate fiber because of acute symptoms, but long-term fiber might actually be really good in protecting. And so I think that could be down to the short-chain fatty acids, yeah.
Okay, thank you.
Our last question.
Yeah, thank you for your talk. My question is with regards to the C-section, because some data has shown in other studies that there is a difference in the long-term effects if it is emergency or elective C-section. Have you in your data looked at if many of these effects might just be, you can say, because of the infant or the not being mature enough, that the pregnancy hasn't been far enough along, that this can be the things that has these effects and not the actual microbiome transfer?
Sure. Yeah. No. We're trying to control for that as much as we can in the animal models, for sure. It's much easier to do than in the humans. In the humans, it's very difficult. What we see in emergency C-section in a lot of the human studies, it's more like vaginally born than what you see in elective C-section. So what we're modeling is some form of elective C-section in our mouse.
Okay. I'd like to join me, please, in thanking John for an excellent presentation.